Okay, so for this lecture, we're going to be talking about the different cellular structures. So basically the cell organelles or the cell parts. As you can see in this diagram, we see what is known as a macrophage or a white blood cell that is eating some bacteria. So you see the long extensions that are called pseudopods, and they reach out and grab the invaders, draw them in, and engulf them through a process called phagocytosis or cell eating, and then they digest them. And this is one of the ways that our body's immune system fights off invaders. So we'll be talking about lots of different things that we can see in this picture. First thing that we see is we see tiny little bacteria. Those are going to be the prokaryotes. And we have one big, large macrophage, which is a eukaryote. So the eukaryotes, the thing that defines them, they have a true nucleus or DNA that is surrounded by a membrane, whereas prokaryotes don't have that. Their DNA is just inside of the cell in what's known as a nucleoid region. Sometimes it's attached to the inside of the cell membrane. And they also have tiny little pieces of free-floating DNA called plasmids, and those carry extra information between the cells. Eukaryotes also differ in that they have other specialized organelles that are enclosed in membranes. And the big thing about this is it allows for compartmentalization or specialization of tasks, which makes them more efficient. And because they are more efficient, they can be larger in size. Things that they have in common, they have their DNA, they have plasma membranes, which is made up of phospholipids. The organelles that they do have are floating in a semi-fluid substance called the cytosol or the cell solution. Sometimes it's referred to as the cytoplasm. And they also have ribosomes, which are important for protein synthesis. Here we have a generalized drawing of a prokaryotic cell. You can see just all of the DNA is just kind of in there, and you've got the tiny little ribosomes. You've got a cell membrane, a cell wall. You've got extensions coming off of that that are known as pili, and that is one of the ways that bacteria can exchange genetic information. It's the equivalent of sexual recombination. And some bacteria have a simple flagella to help them move around. Eukaryotic cells, there are obviously more than just animal cells and plant cells. You have fungal cells, and you've got the protista. But generally, animal cells are going to be without a cell wall. Plant cells will have a cell wall. And animal cells aren't nice and round like this. They all have specific shapes. And plant cells aren't that simplified either. But we're going to talk about all these different types of organelles and their functions. One thing that I do want you to see and recognize is that animal cells, they have mitochondria. Plant cells also have mitochondria. That is not something that is unique to animal cells. Both of them are going to carry out cell respiration to produce their energy. The difference is that plants are going to take energy and convert it to sugars using the chloroplasts, which animal cells do not have. Well, most animal cells don't have. There are some very rare exceptions where animal cells can have chloroplasts. This picture is just a simple illustration about the relative size of eukaryotes and prokaryotes. So you can see that this fragment of the animal cell here is much larger than this bacterial cell, which is much larger than this little bacteriophage, which is a bacterial virus. So this is going to show you how much bigger eukaryotes can get because of their internal specialization that the bacteria do not have. Now, when we talk about cell size, cells do have a limited size that they can attain. And this is because of a ratio between the surface of the cell and the internal volume of the cell. So the bigger the cell gets, the bigger the surface area, yes, but the internal volume is going to grow exponentially larger than the surface area. So essentially what happens is the surface area can't take in enough nutrients and can't get rid of enough waste for the internal part of the cell to survive. So a smaller cell is going to actually have a much larger surface area to volume ratio, which allows that surface area to pull in enough nutrients to maintain the internal environment of the cell. We can see this in this picture. So you have a single cell that is 1 by 1 by 1. Its surface area is 6, but its volume is 1. So it's a 6-to-1 surface area to volume ratio down here. If you make the cell 5 times bigger in its dimensions, the surface area increases to 150, but the volume increases to 125. And when you do that ratio, it's a 1.2 surface area to volume ratio. And basically what this means is the cell can't pull in enough nutrients like sugar and oxygen to go the whole way into the center of the cell before the cell produces too much waste to get out of the cell. So if an organism, instead of being one big cell, is broken up into lots of little cells, then your surface area is much bigger compared to that same volume, and you still have that 6-to-1 ratio. So smaller cells are able to function more effectively at bringing in nutrients and getting rid of waste. Basic cell parts, we're going to go over all of these, and you're going to need to know all of their basic functions. There are going to be some things I'm going to talk about probably in excessive detail, but know your basic functions. All right, the nucleus, this is, what do you want? No, you can't go outside. We're talking about cell parts. Too bad. I know, I'm a meanie. All right, so the nucleus, this is where the DNA is surrounded by the membrane, and different things that you can see. This membrane is a phospholipid bilayer, just like the cell membrane, but it has these pores that are made up of 8 proteins that make these tiny little tunnels to allow stuff to go in and out of the nucleus, because the nucleus has to do stuff, and in order to do the stuff that it needs to do, it needs materials. Here is a photograph where the nucleus has been highlighted with fluorescent markers, so you can see the inside of it. Okay, so inside of the nucleus, we have the DNA that's enclosed by the phospholipid bilayer. We have those pores, which we said takes materials in and out. Things that are flowing into the nucleus are going to be things like ATP. That's going to drive the cellular processes that are occurring in there that gives the energy. The nucleotides for building new DNA and new RNA, and then the enzymes that do all of that work. Those enzymes are made in the cytoplasm, and then they have to come into the nucleus to do their jobs, like copying the DNA and making the RNAs for protein synthesis. Things flowing out of the nucleus are going to be ADP and phosphate. So when the ATP is used, it breaks down into ADP, adenosine diphosphate, and the third phosphate is broken off. That gives the energy for the cell processes. So then these two things go out of the nucleus to be put back together by the mitochondria to allow cell processes to occur. The ribosomes are made in the nucleus and then they are shipped out. RNA is also made in the nucleus and shipped out, and both of those together are going to make proteins for all of your cell processes. Other stuff, DNA is in there. That is organized into loose strands called chromatin. That's the normal way that DNA is in the cell. When the cell is undergoing cell division, that chromatin will condense into chromosomes. They condense around proteins called histones. The histones are also going to be important for determining which genes in the cell are actually expressed. So you've got all 46 chromosomes, you've got the 20,000 to 30,000 different genes, but not every single gene is expressed inside of every single cell, or all of your cells would be identical. So maybe 2 to 3% of the DNA in your cell is going to be expressed, and it's these histones that help regulate which genes make it a liver cell, which genes make it a muscle cell, which make it a brain cell. So that is part of the process called cellular differentiation, so that each cell can do its particular job. All right, so then when it's time to divide the cells into two new cells, it is much easier to organize those loose strands into condensed structures, and that's what the chromosomes are. So imagine having 46 strings in a pile and you were trying to separate those. It'd be much easier if you coiled them up into 46 little balls, and then it'd be much easier to separate. That's what the cell does with the DNA. So this is the way that DNA is organized around histones. It is loose. This is the chromatin over here. And then it's going to wrap around these histone proteins, and then those histone proteins connect together, and then those connect to another protein fiber, which causes them to coil, which then causes them to turn into the chromosome. That's all one of the ways that your cell, which has 6 feet of DNA, all of that can fit into something that is much smaller than a typical period on a page. Inside the nucleus, we also have an area called the nucleolus. It's not really a structure, but inside of the nucleolus, you have concentrations of proteins and what is known as ribosomal RNA, and that is going to be where ribosomes are made. And we'll talk about ribosomal structure in a moment. So these ribosomes are made into two parts, and then those are shipped out of the nucleus into the cytoplasm, and then they come together to make proteins. Ribosomes, as I said, are going to be the site of protein synthesis. These are going to be made in the nucleus. They are exported into the cytoplasm. They're made of RNA, specifically they're made of ribosomal RNA and proteins, and they are organized into two parts. One is bigger, called the large subunit, and the smaller one is called the small subunit, and they come together to make proteins. And these are going to be slightly different in prokaryotes than in eukaryotes, and that makes them a target for antibiotics, for some types of antibiotics. And since our ribosomes are different, they aren't affected by the antibiotics, so our cells can continue to make proteins and survive, and because the proteins can't be made inside of the bacteria, they end up dying. And this is some basic pictures of, this is your large subunit and your small subunit, and different more complex pictures here, but this is the typical one that you will see in textbooks. And they exist as separate pieces, and then they come together And then in between them right here is where the RNA goes through and it builds off proteins in the protein synthesis process. Now inside of the cell, there is a manufacturing, shipping, sorting, and repackaging system that is known as the endomembrane system, endo for inside. And this is a system of tubes and vesicles that make stuff and ship it throughout the cell for important complexes inside of the cell and to ship things out like enzymes and neurotransmitters and things like hormones and things like oil and sweat. So it's basically an assembly line. Some of these membranes are in direct contact with each other, and some of them communicate by sending little shipping packages known as vesicles from one to the other, and so they can exchange things. So the one membrane kind of finishes its job and pinches off and forms this little sac, which is called a vesicle, that migrates to the next section of the factory, and it merges, and then that stuff inside of there gets changed in a complicated process. So what we see up here is our nucleus, which is connected to the first part of the endomembrane system, which is the endoplasmic reticulum. There's the smooth ER and the rough ER. And you can see here a little vesicle pinching off. It has some protein components inside of it, and then that gets shipped to the next part by that vesicle, which is the Golgi apparatus. The Golgi apparatus is going to kind of process and kind of fine-tune those products, and then it will make a new vesicle at the end, and that vesicle will either stay in the cell and do a specific job, or it will merge with the cell membrane, and whatever's inside of it gets dumped out. This is how your pancreas makes enzymes and then secretes those into the small intestine. This is how your nerve cells make neurotransmitters that then release and then they cross the gap between two neurons to stimulate the next neuron to send the signal on, which we'll be talking about later. So this is your basic flow. Nuclear envelope to the ER by a vesicle to the Golgi, and that is either going to make a lysosome, which stays in the cell, or a vesicle for secretion. Endoplasmic reticulum comes in two types. Reticulum means network, endoplasmic inside of the cytoplasm. And these tubes and cisternae, the cisternae are simply the ends of the tubes. Think of it as a cul-de-sac inside of a neighborhood. The tubes are the roads that go through the neighborhood, and then the cisternae are the cul-de-sac. So imagine a bunch of cars drive up the road, park in the cisternae or the cul-de-sac, and it pinches off, and then it moves to another neighborhood, and then it merges with a road over there, and then those cars can drive through the next neighborhood. That's essentially what's happening, except instead of cars, it's proteins. So here is our system. And one of the things that I want you to see in this picture is we've got our product, which is this little star right here. And that little star is made inside of the endoplasmic reticulum. And then you've also got this surface protein, this thing that looks like a yellow peanut. They merge, and as they're going through the Golgi, through this whole process, that protein will get changed, and we're just going to mark the outside of the peanut with this green star. You know what? We're going to make that a purple star. No, that's already purple. We'll go red. A red star. So that's right here. And when this merges, the that red star is on the outside of the vesicle, but when this membrane merges, that red star is on the inside of the cell. And then the interior purple star, which is on the inside of the vesicle, when that merges, that goes onto the outside of the cell. So whatever's on the inside of the vesicle becomes the outside of the cell, and whatever's on the outside of the vesicle is going to be on the inside of the cell. So when it kind of inverts. And so if this were filled with, say, digestive enzymes, those would be then released out into the outside of the cell. Okay, so our types of ER, we've got the smooth ER. They are smooth because they do not have ribosomes attached to the outside of them. Main functions of the smooth ER is for lipid synthesis, so making fats, making oils, making new phospholipids for the cell membrane to grow. Because if you've got a small cell, it needs to grow and get bigger, so you need more cell membrane. And also steroids such as hormones are going to be made here. Smooth ER also process carbohydrates. So when you eat a bunch of starch, your digestive system breaks it down into glucose, which goes into your blood. And then if you've got too much glucose in your blood, your body needs to store it. So the smooth ER inside of your cells can convert that to glycogen to store it for later. That mainly happens in the muscle and in the liver. Speaking of the liver, one of its main jobs is to detoxify the poisons that enter into our body. And this happens in the smooth ER of the liver cells. So things like alcohol and medications are going to be metabolized in the liver by the smooth ER. This also stores calcium ions. This is mainly going to be happening in muscle contraction. And when we're talking about the smooth ER in the muscles, we're going to call it the sarcoplasmic reticulum, because the contractile unit of the muscle is called the sarcomere. And so instead of the smooth ER of the muscle, or the smooth ER of the sarcomere, we just call it the sarcoplasmic reticulum. We'll get back to that later. This also forms a structure called a peroxisome, which we'll talk about later as well, but this is something that makes and gets rid of hydrogen peroxide, and we'll talk about why that happens. So quickly, we see our nuclear envelope here. These are the nuclear pores, and then your DNA would be inside. This would go the whole way around, be a sphere or whatever shape it is. Here is the smooth ER with nothing attached to the outside, and then down here you can see the little red dots, which are the ribosomes, making it the rough ER. All right, so the rough ER then, this is the ones that have the ribosomes attached, which are going to be for protein synthesis. Now the rough ER does not have permanently attached ribosomes. They only attach when they are making a protein that's going to be part of the cell membrane or inside of a vesicle. We'll talk about how a ribosome knows it's supposed to do that later. So these proteins will either be secreted from the cell or they will become part of a lysosome, which is for digesting things inside of the cell. So as the ribosome attaches to the ER, then the protein goes in through a pore. And then once it's in there, it may get extra things attached to it to give it specialized functions. And some of those things are called oligosaccharides, which are those short sugar units that can make them stickier, have different attachment sites to cells. Once they are made, the ones that are going to be secreted, they are going to then go through the Golgi apparatus, and they'll be shaped by the chaperone proteins until they are ready to be shipped out. The way that a ribosome knows to attach to the ER has nothing to do with the ribosome. What happens when the protein is being made, which we can see right here, this is the protein being made by the ribosome, there'll be a little signal protein at the beginning, which attaches to something known as a signal recognition particle. Then that attaches to this receptor on the surface of the ER, and then that causes this pore to open, and so the protein can then enter into the ER. So it's all about this complex communication signal. And then once that's done, once this protein is made, that ribosome will detach and attach to another messenger RNA, and it might make a cytoplasmic protein and not attach to the ER again. So the ribosome doesn't care what it's making. It only goes there if the protein it's making tells it to. Rough ER is also going to have the job of making new ER. So if you think about what the rough ER is doing, at the end it's pinching off a vesicle and sending it to the Golgi. So that means every time it does that, you lose part of the rough ER. So new ER has to be made so that process can continue. So that means that the rough ER has enzymes in the membrane that are going to then help it make new endoplasmic reticulum. So that's all extra stuff that you don't really need to know. Just know it makes new ER. So the vesicle pinches off, goes to the Golgi apparatus, and it's just this series of flattened sacs, and it's going to accept those vesicles. The stuff that's inside that enters into the Golgi and kind of moves through like a factory, and in each step something happens to that protein to change its shape and therefore its ultimate function. The end that receives it is called the cis end. It moves through, and then the end product comes off of what's known as the trans end, the opposite of where it came in. And then from that shipping end, it either stays inside of the cytoplasm or it gets shipped out. So here is your incoming vesicle. So this would be the cis end. The proteins move through this process. A vesicle is pinching off here, and this is going to be the trans end. And then this vesicle that's formed does its job. So the Golgi apparatus is going to do lots of things. It's going to alter phospholipids that are going to become part of the cell membrane. When we get to talking about the cell membrane in particular and in depth, we'll talk about why phospholipids may need to be different. The outside of the cell is called the extracellular matrix. That's what helps hold cells together. So the cell glue is made by the Golgi apparatus, and then it sorts those products that needs to be secreted. One of the things made is a lysosome. The lysosome is a organelle that stays inside of the cell, and it is filled with these digestive hydrolytic enzymes. And so hydrolytic means hydrolysis, so breaking things apart. And inside of it, it has a pH of 5, which is much lower than the regular components of the cell. And this actually is important because if the internal cell environment is getting too acidic, it can pump those extra hydrogen ions into the lysosome to maintain the pH. And so what happens is the pH of the lysosomes, the hydrolytic enzymes are specific for that pH and function best there. So that helps them. If one of those breaks open, it's not going to go and kill the cell. But if a bunch of them break open, then you can enter into something that's known as apoptosis or programmed cell death, which is actually pretty important and really, really fascinating. All right, but its main job is if the cell brings a bunch of food in, then it will make a food vacuole, and that food vacuole will merge with the lysosome, and it will digest everything inside for the cell to use. All right, so the cell brings in, so if it is that macrophage, that white blood cell on that first slide, it brings in those bacteria. it brings it into a food vacuole. That food vacuole is then going to merge with the lysosome, and it will digest them and kill them. The cell also is going to get rid of old and non-functional organelles. This is called autophagy or self-eating. And then once the lysosome is done digesting those old organelles, it can reuse those molecules to build new ones. And then one of the things that lysosomes do, if a cell starts going wonky, then apoptosis, or that programmed cell death, can be triggered, and that non-functional cell can be destroyed. So here we have food coming in. You see it pinches in and forms this vacuole, and then it merges with the lysosome, and that digests those food, and then those can be used by the cell. A defunct mitochondria here that merges with the lysosome, and we can recycle. Okay, so apoptosis. I'm going to talk a lot about this. What do you need to know? It is an important mechanism for organisms to get rid of non-functioning or damaged cells. It's important for changing and developing embryos. And it sounds bad, but it's actually very good. All right, so the lysosomes release these chemicals that are called capsases, and these are protease enzymes. So protease, these are enzymes that break down proteins. And basically when these release all of these enzymes, then it basically breaks down the internal structure of the cell, breaks its internal framework apart, and the cell begins to break apart into these tiny little pieces that macrophages and your immune system can come along and eat and clean up. So one of the main things about apoptosis is that in our bodies, we're going to lose about 50 to 70 billion cells a day by this process of apoptosis. These are old and worn out cells that need to be destroyed. And a lot of these are actually immune cells. So as our body is making new white blood cells, they go through a process called self-tolerance testing. One of the important things about our immune system is that it recognizes tissues, invaders, bacteria, viruses that don't belong in our body. So those are things called non-self cells. They have a different exterior surface, different proteins, so they have a different cellular and molecular identity on the outside of the cell. Our white blood cells are trained to recognize those non-self cells and destroy them. The unfortunate thing is the production of white blood cells is basically random, and a lot of the white blood cells that are produced are going to recognize our own cells as non-self. So that's what this tolerance testing is. If a white blood cell recognizes our body tissues as the enemy, that's not good, because that could get out into our body and it could start attacking our own cells and destroying them, and that's what's known as an autoimmune disease. So the self-tolerance test, if a white blood cell fails that, it needs to be destroyed. And that's what we mean by an incorrect immune cell. That is a cell that would look at your liver and go, oh, that's bad, let's attack it and destroy it, which we don't want because we want our liver to be healthy. It's also estimated that we kill about 5 to 6 cancer cells every day. That doesn't mean you qualify as a cancer survivor. People that actually are cancer survivors would probably find that a little offensive. And this is how the menstrual cycle initiates. So as the interior of the uterus grows and gets ready for an embryo to implant during the beginning of the menstrual cycle, if pregnancy does not occur, the lining of the uterus, the endometrium, is going to shed, and that's what menstruation is, or the period. And this apoptosis is the thing that initiates that. And we will be talking about the menstrual cycle in depth much later. Also, so cells that are wearing out, cells that have been damaged by radiation or chemicals, are all going to hopefully undergo apoptosis. This is also important for the developing of tissue. So you can see here's a little baby that's just been born, and you can see that these digits did not separate properly. So when you are developing, your hand is basically like a flipper. But apoptosis helps separate those digits. If that does not happen, that is called syndactyly, where they are fused. And so that helps us develop properly. Caterpillars into butterflies, so when they go into their chrysalis, their body gets broken apart and then rebuilt. The disappearing of the tadpole's tail is by apoptosis. And in plants, after a flower is pollinated, the flower has done its job. It has brought the pollinator in, it has been fertilized, so maintaining that pretty structure is going to be metabolically expensive. So once the job of the flower is done, let's get rid of it, stop wasting those nutrients and resources. So apoptosis gets rid of that flower, so then the plant can concentrate on developing the seeds and the fruit, so it can continue its reproductive process. Unfortunately, lysosomes don't always function properly. One genetic disorder that results from dysfunctional lysosomes is Tay-Sachs disease. And this results when the lysosome has an improper enzyme that is called hexosaminidase, and you don't need to know that, but that is for digesting lipids. And if this enzyme is non-functional, the enzymes begin to accumulate in the brain and cause severe neurological damage and mental functioning, and eventually the individual with that will die. This is typically found in specific cultures, specifically Ashkenazi Jews, Cajuns in Louisiana, and then in some groups of French Canadians. The Ashkenazi Jew and the Cajun mutation is actually the same mutation, but the French Canadian mutation is different. So this is the same disease from two different mutations, which makes it even more interesting. That is one type of genetic disorder. Vacuoles can be formed from the endomembrane system. These are tiny little storage vesicles. They can store proteins or enzymes. And what happens is vesicles confuse together to make vacuoles. The main vacuole that you need to be concerned about is going to be the central vacuole, but in animals, it's mainly food vacuoles made by phagocytosis. You also have what's known as a contractile vacuole in small protists. So we have a complex system called the urinary system to get rid of excess water. Individual cells, such as a paramecium, does not have any complex organs, so it has these vacuoles that absorb excess water. They merge with the cell membrane, and they get rid of the excess water. It's their urinary system. The central vacuole in plants, think of that as an inner tube inside of a tire. You inflate the inner tube and it makes the tire firm and rigid to give it the car support. Well, the central vacuole in a plant cell fills up with water, pushes against the cell wall to give the plant cells support. So when a plant doesn't have enough water, the central vacuole gets less firm, or less turgid is the correct term, and then the cell walls start to collapse, and that's what causes plants to. So the main functions of the central vacuole is really, again, to support the interior of the cell wall but it is also for storing other things like metabolic wastes and pigments and ions. You can see in this picture this empty space. It's not empty, it's filled with water. That's the central vacuole, and that's pushing out, giving support to the interior of that cell. Some of the things that can be stored in the central vacuole are toxins that actually make the plant poisonous to things like insects and herbivores, or they might just taste bad, or they could be filled with neurotoxins like nicotine and caffeine, which are toxic to insects but have very different effects on mammals. So this is that central vacuole pushing out against the wall. The membrane of the central vacuole is called the tonoplast. Do you need to know that? Probably.